I'll be showing a bunch of data that was generated primarily by Sean Wan, a senior graduate student in the program, and work that involves a collaboration with Andres Perez and Mohammed Al-Khamis. I'll be stealing some of Andres's thunder, which I apologize for in advance now that I saw his presentation this morning. And I want to point out Bob Morrison, whom you all know. Bob has had an enormous, profound influence in the swine industry by teaching people how to work together and share information. And these are the producers and organizations that are now sharing swine health and disease information for the entire industry, and it's a model for the world. So we're going to talk about PRRS genetic diversity and evolution, and we'll give a story about the 174 virus. There are two general types of PRRS viruses, as we're familiar with. Type 1, which was known as European and is primarily in Europe. Type 2, which was discovered in North America and is the dominant virus in North America and Asia. And both of them have a lot of genetic diversity, shown here in OR5, and there's nothing in between, which seems to be an astounding phenomenon, but it tells us something about how the virus evolved. So the first issue here is, how do we talk about PRRS viruses when we describe genetic diversity? Fred Lung came up with the idea of referring to genetic lineages, a common sense idea, and he took all of the known PRRS genetic diversity known at the time and generated a dendrogram which revealed families of viruses and called them lineages. 5.1 is special because most of the current 5.1 lineages are derivatives or field reisolates of the Engelvac MLV vaccine. Lineage 5.2 are viruses, the oldest of which are clearly related to VR2332 and Engelvac MLV, but have a distinct genetic origin. I want to point out IF CLOP from Canada, lineage 1 founding virus discovered by Serge Day. He was an enormously positive influence in the early days of PRRS virus in Quebec, and Carl Gagnon, one of his students, is following in his footsteps now. But Fred pulls his hair out because we don't talk about lineages when we talk about PRRS viruses. In the U.S., we talk about RFLP patterns. And the main reason for this is that Ron Wesley figured out a simple naming system in the early days of PRRS that led to the clear identification of the 252 as being unique from all other PRRS viruses. So it was an early marker that told people that they had the vaccine. And you can see that in general, the 184 family of viruses is in lineage 1, the 174 is. 142s have been around forever and are in lineages 8 and 9. The high-path PRRS, as I think up here somewhere. But we've got two different naming systems. And one of the problems here is that these are all based on ORF5. So they're not very useful if you're looking at ORF7 in Europe, for example. Another way to look at the viruses is by whole genome sequencing. Now, we're a long way from generally using whole genome sequencing as a way of characterizing PRRS viruses, in contrast to ORF5, where there are probably 20 or 30,000 sequences hidden and known in the world. So you've got where you have this enormous database where you can really find things that are closely related to your virus. For whole genomes, there's only about 300. And more than a third of these are almost all the same. China has lots of high-path PRRS, and Chinese scientists love to sequence it and put the whole genome into GenBank. it kind of obscures the fact that there's an enormous genetic diversity of type 2 viruses in China and in Southeast Asia, probably as many as the diversity is as great in the US and Canada. The other big group is the Engelvac vaccine reisolates, and then you see a scattering of other things. And if you tried to put assign lineages to these, it would be all mixed up because there's a major amount of recombination in PRRS virus. So GenBank, if you use GenBank as your source for looking at diversity, what you're really looking at is the biases of scientists at what they want to study. And there's no good naming system for genomes. Okay. So just a little bit about the way RFLP came about. There are 3 restriction enzymes that digest once or a few number of times in the ORF5 600 base sequence, and depending on the pattern of where restriction sites exist in this sequence for these 3 enzymes, the naming system was developed. And when there were only 20 sequences known, this worked great. By 2010, it was getting awkward because there were 4 different MLU1 forms, 55 HINK2, and 8 different SAC1s. But they still had a lot of utility at the local level. If you had a 144 on your farm and 3 months later you got a 144, sequencing would almost always show it was the same virus. If you got a 252, the vaccine had come in there somewhere. If you had a 173, that was an outside introduction which had different implications for your ability to control the virus. By March 30, earlier this year in 2015, we were up to 114 different HINK2 sites and 12 HINK3. And the most recent email that came out from the VDLs that keep track of this stuff a couple weeks ago, we now have 1 more MLU1 form. We've got 125 in the second position and 13. This is kind of unwieldy. And it's especially unwieldy since the RFLPs do not infer genetic relatedness. And that's obvious if you look at this example with 144 RFLP types showing up in very widely divergent clusters of genetic similarity in a set of sequences basically taken from one system or one veterinary clinic. Okay. So I guess because the veterinarians and producers in the field are the ones that we're trying to translate our information and knowledge to, most of the talk will relate to what they understand, which are RFLP types. And here we're looking at variation over a 10-year period in the abundance of different RFLP types sequenced at the Minnesota Veterinary Diagnostic Lab. So in 2001, the sequencing was completely dominated by, I thought it was it must be the 142s. And then at the end of 2001, the 184 strain showed up, and these are fit viruses that compete effectively in the environment and became very abundant. And then they started to decrease at times that new isolates were appearing. This, I think, is the ATP vaccine, but down here we've got a new family of 118.2s and then 126.2s. These are progeny of the 184 family. So we see that the distribution of viruses, the virus types in the environment of commercial farms is changing. One of the questions we had was, I guess a scientific way to put it is to ask the question if the genetic structure of PRRS viruses in the field is reflected in what you see in GenBank, or is it different? And we were able to address that question because Bob Morrison got U.S. swine producers to give us access to their sequence databases that they had accumulated in response to trying to figure out what PRRS was and how to control it and what was working. So starting in 1998, these large systems were routinely sequencing PCR positive samples, usually from outbreaks, sometimes as a result of active surveillance, and just accumulating them in their databases. By 2015, these 5 producers had almost 7,000 sequences, almost none of which were in GenBank. And if they were, you would not be able to tell because they'd been put in with different names. What we see is a whole bunch of type 2s, and 1.5% are type 1 viruses. So they're present. I regard them as insignificant. But what I thought was really neat was you had these little lines here and here and all of this and this. These are a whole bunch of isolates that have the same sequence. So this is a picture of PRRS virus evolution that's completely contrary to everything that's in the literature, which says that this is a really rapidly evolving virus and it's changing all the time. It's not changing all the time. One of these is the MLV vaccine. It doesn't change because the same thing keeps getting put in for 20 years. So when it comes back out, it's still about the same. But these others are virulent field viruses that for a period of time are popping up many, many times, and they're not changing. So we don't know exactly what this means, but it tells us that the mechanisms of selection and evolution probably are not just random mutation that would give you gradual change, that it's more likely some kind of saltational effect that is more consistent with large-scale recombination being a major driver. So things do not change until you get a recombination. This pattern of maintenance of a consistent, similar sequence over time also characterizes the high-path PRRS viruses in China. So this is a virus which evolved somehow from a pre-existing endemic strain in China to very high virulence, and over a period of years here where we're looking at more than 5 years. Here's 2012, mostly 8 and 9, 2012, 2009, and so this virus is not changing very much, surprisingly little. So this phenomenon is not unique to the U.S. or these 5 systems. So now I'd like to look at one system which over a period of years collected about 160 field virus ORF5 sequences, and to try and understand what they were, a set of reference strains was put in. These single deep lines going out here are the references, and the rest of these are the field isolates. And what we see if there was one type 1 virus in there. If we take that out, where it runs off the page here, we get a higher resolution image, and one of the interesting things is there's 2 sets of sequences which show very little change. This one and this one, where there are probably 70 or 80, well, you can count the number here. A bunch of these are identical. It's the same sequence, and you've got some here where there's 1 base difference or 2 bases difference. So this is not changing. It turns out that this little group was from a 144 RFLP outbreak that started in April 1st, 2012, and the last sequence was isolated June 17th, 2013. And you can see that Not very much genetic variation here, and they're pretty much all 144s. They're just not changing very much. But if you think about it, this is a virulent virus that got into a large system that was spreading in the system, and then it stopped. Why did it stop? Well, we've got two ways of controlling PRRS virus: vaccination and herd closure, and methods based on those. They work. Oops, got to go back one. Thank you. Even though this is a highly virulent virus, it succeeded in getting into the herd and spreading for a while. We got control of it. They. Come on. Okay. So it says that this didn't just die out, and it didn't continue to spread, even though it had these virulence characteristics. It was not going to be a huge problem. Now we need to go back again, because I forget which one's the pointer. Okay. So this big group is the 174 viruses, which appeared in this system May 21st, 2014, and less than a year later had broken in 25 of the 27 sow farms in this system. And you see a bunch of 174 viruses here. You see very little genetic variation, but I try as best as possible to refer to these as clusters or families, because they're not all 174s. You have to be careful about looking at RFLPs to tell you what you have. One base change can change a 173 to a 174, and on and on and on. Okay. So this 174 virus in this system at the same time also was in other systems. So in this 5 systems, 241 of these 6,700 viruses were in the 174 family, shown here. And we've got 48 other nearest neighbors here from this entire sequence. So the nearest neighbors are right here. And if we look at these, we see that these are progeny of the original 184 family that showed up in the U.S. in 2001. It showed up in Canada when Serge Day did his first sequencing in around 1991 or 1992. So it's a virus that's been in North America forever, and its progeny. Okay. So this is clearly a highly virulent virus. It has biological properties that we're not very familiar with. It appeared to first show up in I think the first case was in Indiana or Ohio, and over a period of a few months it showed up in other locations, and the colors correspond to different genetic groups, but these are not very different. This distance here is 1 base per 1,000, so about double that length is 1 base change. So again, very few changes, but large geographical distribution. And here we're looking at the sequences of those early viruses. This is the rectangular dendrogram you're accustomed to looking at, and you can see that there are not very many differences here. And if you take the bottom here and made this flexible and twisted it around, come on here, you'd end up with a circle as shown on the right side. And the heavy blue line shows all of the 174 viruses in this example. Okay. Looking at this nucleotide sequence of this virus has not been revealing. It looks like a virulent PRRS virus. So Here we're looking at a list of viruses, and this is the NSP2 region, the 5' end of the open reading frame, and you can see where deletions occur. The high path Chinese PRRS is right here. Here are its deletions. Those same deletions show up in our 174 example here. It also has this deletion that is characteristic of essentially the entire 184 family and all of its progeny over the years. But if you look carefully, you see that it's not the same. There's a little more sequence on each side. This region is not responsible for the virulence of high-path PRRS viruses. Yanhan Chun in Beijing did a gorgeous genetic experiment several years ago where he swapped the NSP2s out from the virulent high-path PRRS and the avirulent form and put them into the opposite background, and with the hypothesis that virulence would go with the NSP2 phenotype. And it did not. It stayed with the backbone. This is a very interesting region of the genome because it's so plastic. It tolerates lots of deletions, which says that those sequences taken out are not essential. It allows people like Kay Fauberg at National Animal Disease Center to look at putting foreign sequences into this region as making PRRS virus a potential vaccine vector carrying other antigens or other markers for vaccine use. But in terms of virulence and pathogenesis, no one has really been able to show that with good evidence that the virulence is associated with all this variation. Also, if you look at a large range of 174 isolates shown here, there's very little protein variation in GP5. We're looking at ectodomain 1, transmembrane region, ectodomain 2, the exterior parts of the protein that could be susceptible to immunological selection, but we see really no significant change at all. The hypervariable glycosylation site here is essentially unchanged. Let's see, what do you call it here? The MK. There's another typing system that looks at this region right here and shows a lot of variation, but we see very little there as well. So no evidence of directed evolution or selection during this outbreak. Okay. Having access to these really large databases where all of the sequences are being saved allows us to start asking questions now that Andres Perez will go into in much more detail in his presentation. But I will lay the groundwork, I hope, for that by showing some of the things you can do with not only having the sequences, but also having the locations where they were isolated, the date of isolation. These producers know the day the sample was taken. They know the longitude and latitude to 8 decimal places where they were found. Really, and many of them have all of the clinical information associated with the outbreak that they were taken from. So these are really rich sources of information. Statistical phylogenetic methods using Bayesian approaches can incorporate this epidemiologic information into genetic analyses and give you pictures of how viruses changed over time. You had this amount of genetic diversity, and when the outbreak occurred, you had this explosion of viruses and this increase. You also get probability estimates. This is the average line, but you can see it could be anywhere in here. So these are very powerful methods. You can ask Since you know where the viruses were found, you can ask how they move geographically. And so you can generate information here. You can look if you have information on where in the system they came from. Was it a farrowing room? Was it the sow barn? Was it a finisher? You can ask if the outbreaks are isolated in a particular production type. And you can look at change over time. This is a very nice little movie, and I'll let Andres show it. And it's clear that this virus moves around. You saw many slides ago where early isolates of 174 were distributed from North Carolina to Iowa. Well, I've been saying for years that pathogens go where their hosts go. Viruses go where their hosts go. PRRS goes where pigs go. Pigs go from North Carolina to most of the rest of the U.S., especially Iowa and Minnesota. So it wouldn't be a shock if viruses in North Carolina went to those regions. In Minnesota, pigs go from all over the country to Minnesota. Okay? So it wouldn't be a shock if new viruses showed up in Minnesota that no one knew about simply because they were somewhere else in the country and those sequences were not publicly available. They're only seen when you get here. These kinds of questions become relevant when we talk about large-scale PRRS control systems, where you're trying to understand how to control virus outbreaks, and you need to know if these are endogenous viruses that are escaping current control practices, or are these viruses moving in from somewhere else? Well, it helps to know what the viruses are somewhere else. So if the virus shows up, you can ask where that was. Can you implicate pig transportation as a route? That's not to blame people. That's to say, well, your transportation cleanups need to be much better, or you have to pay attention to where the trucks go. You don't want them driving through a cloud of PRRS as they go through northwest Iowa, just to throw something out there. So take-home message is large-scale PRRS outbreaks have occurred frequently in the history of type 2 PRRS, and we can get more information than we had in the past as we start to see these very large databases. And I might point out that these producers are allowing their sequences to be deposited in GenBank, not with longitude and latitude, but geographic location, year of isolation, and it will provide a new level of richness for Fred Lung to mine. He's nodding his head, yes, I can't tell how excited he is. Genetic change during outbreaks looks to be limited. That's new knowledge. And what are the implications of that? Bayesian phylodynamics helps understand disease spread, and PRRS virus goes where pigs go. So in terms of really understanding the PRRS-pig equation, it would be helpful if we understand pig movement in greater detail so we can understand the nature of outbreaks and how to intervene. Thank you.